I'd like to echo Greg's thanks to the organisers. It really is a pleasure to be here. Surprisingly enough, despite Greg and I having worked so closely many years ago, we've hardly ever spoken together at the same conference. And I didn't know what he was going to talk about today. And I'll be following up many of his points. And I have to thank, again, the organisers for the way they've produced the programme, because I naturally dovetail between Greg and Pernilla, who's following me. I've been very fortunate to have had a very long career in protein science. In fact, it spans 7 decades. I started my work in the 1960s at the time of the first protein structures being determined by X-ray crystallography, many of them at the MRC Laboratory of Molecular Biology, where I worked for much of my career, and most of my time I've been funded by the Medical Research Council, even when I wasn't there. When I started, the protein folding problem began to be formulated. And this problem is composed really of 3 separate parts. It was begun by Chris Amundsen, who showed that denatured proteins could naturally and spontaneously refold to their native structure. And people started to wonder, could we predict the final folded structure of a protein from its amino acid sequence? Over the years, we've accumulated tens of thousands of structures of proteins experimentally. And this year, the Nobel Prize was awarded to the inventors of AlphaFold for finally being able to solve that problem and predict structure. The next part of the protein folding problem is there a simple code that directs the folding of a protein to its final folded structure? And the answer is, we don't know that. And the third part of the problem is, what is the mechanism of protein folding? Well, kinetics and mechanism basically are my passions. And we're getting somewhere in this direction, and I'm going to be talking about that today. And what I want to do is to go back to very first basic principles of chemistry that I don't know how much people know, and show how it's developed over the years, and to use technologies of discovering and proteins and methods that have been developed over this period of time. The protein folding mechanism problem was first formulated by Cyrus Leventhal in 1969. At a conference somewhere or other, he pointed out that if a protein, even a very small protein of 100 amino acid residues, folded purely by random motions of the protein, it would take longer than the age of the universe for that protein to fold. So it's clear that a random search can't be taking place. So the theoreticians and some experimentalists went away, and they came up with possible mechanisms of how a protein could fold. And in complex problems, one way of solving them is to break them up into simpler processes. So on the one hand, people started thinking, but I think this is really due to a Russian scientist, Petitsin, that perhaps a protein could fold in parts by the secondary structural elements like alpha helices and beta sheet forming rapidly, and then the elements of secondary structure would then dock to form the final folded structure. Leventhal himself thought perhaps proteins could fold by a classical nucleation growth mechanism, where perhaps this element of secondary structure formed very slowly, and then growth would occur very rapidly from that nucleus. And finally, a third mechanism of hydrophobic collapse, also proposed by Petitsin, was that perhaps proteins could just collapse with the hydrophobic interactions, making it form a ball, and then it could rearrange more rapidly within that. Early experiments then were conducted in many labs on protein folding kinetics, and it seemed that protein folding always passed through intermediates on reaction pathway, which would accumulate. And that meant that the nucleation mechanism went out of favor, because a nucleation mechanism has a slow first step followed by a very rapid growth step, and so it means that any intermediate that's formed is present only at very, very low concentrations and wouldn't be detected. Why do we really want to study the kinetics and mechanism of protein folding? Well, quite simply, it's the intellectual challenge of solving the problem of knowing how a protein folds and gets around the basic problems of a random search. But very importantly, it's to understand as well the role of protein folding in disease. From Alzheimer's to cancer, proteins can unfold, misfold, and refold, and we want to learn about that in order to develop methods and technology for tackling those diseases. So what I'm going to talk about today is my initial program on studying protein folding, beginning on the lines of the first experiments with Greg. And I wanted to measure experimentally the interactions that held proteins together and responsible for protein stability. And using procedures I'm going to talk about in the next few slides, were to make modified proteins to understand the mechanism and really try and get things done at an atomic level, and then collaborate with theoreticians to have experimentalists and theoreticians get together to be able to predict things. So the problem is, if you're doing this experimentally, as a protein folds up, it passes through a myriad of intermediate states and making and breaking noncovalent interactions. And we want to be able to measure these as the folding process proceeds. Now, I had a background in classical physical organic chemistry of solving mechanisms. And by chance, it was the first compound on Greg's slide. I looked at the hydrolysis of aspirin for my PhD. And the way we do that in classical chemistry is we have to characterize all of the ground and transition states in a reaction. As a reaction proceeds, a reaction can go through a transition state, which is at the maximum in a diagram of free energy against reaction coordinate. Transition states are unstable and don't accumulate. Then intermediates can be formed, and intermediates are at the bottom of little valleys in free energy profiles. They can accumulate, and we can study those directly on occasion. But to understand mechanism, really, one has to characterize these transition states. And there are basic equations relating the rate constant of reaction, K, to the free energy of activation, delta G, double dagger, and with some constants in between. So if we can look at a transition state and try and make guesses about its structure and how the structure would be sensitive to changes in structure elsewhere and environment, we can dictate how a reaction proceeds, or we can analyze the transition state in reverse. How do we study transition states? Well, this goes back to, I think, my biggest hero in science. It's a Danish chemist by the name of Brønsted. He was, I think, a very great man. He was an exceptional scientist. In Denmark during World War II, he was an active anti-Nazi, and on the basis of that, he got elected to the Danish Parliament, but unfortunately died before he could take his seat. Some people might have heard of him because they've heard of the Brønsted-Lowry theory of acids and bases involving proton transfer. And he founded my area of physical organic chemistry, where I started, and it's the 100th anniversary of his most important paper in this. Back in 1924, along with Pedersen, Brønsted discovered a phenomenon of general acid-base catalysis, where acids and bases can catalyze reactions. I was interested in this. Like Greg, I was interested in the mechanism of enzyme catalysis and acids and bases at the active sites of many enzymes and help catalyze the reaction. What Brønsted and Pedersen did was to show that a base, such as the acetate ion, could enhance the attack of water on another compound like an ester. So an ester would be relatively stable in water, be rapidly hydrolyzed by hydroxide ion, but in the presence of something like acetate, the acetate ion will speed up the reaction. And the way it does that is that the negative charge on the acetate ion would cause the partial transfer of a proton from water to make the water more like a hydroxide ion, and then the hydroxide ion at the same time will be attacking the carbonyl carbon of the ester. And so there's partial transfer of a proton from acetate to water and attack on the bond. And what they discovered was that the stronger the base, that's the catalyst, the better is its catalytic power. And they found that there was a linear relationship between the logarithm of the rate constant for this catalysis and the pKa of the base. And of course, the pKa is also a logarithmic measurement. So there's a little equation, log K2, the rate constant, equals some number times a constant, which became known as beta for base, but it's really beta for Brønsted nowadays, times the pKa. And that translates into free energy terms. We know that the rate constants for reaction are proportional to the free energy of activation, and that is proportional to the free energy change in the actual reaction itself with this Brønsted beta. And beta is defined by the slope of the curve, or if you just take 2 points, the difference in free energy of activation divided by the free energy difference in the equilibria. And a linear free energy relationship like this is an example of generic ones which have been found for many different reactions over the years. The importance of beta is that beta, Brønsted's beta, is an approximate measure of the degree of bond formation in a covalent transition state. If there's no transfer of that proton, for example, the change in free energy of activation on change of base is 0, and the Brønsted beta is 0. It's 0 divided by a number. If there's complete transfer of a proton during that reaction, it's like the full ionization of that acid or base, and you get a Brønsted beta of 1. And for partial transfer, you get partial values of beta. So beta of 0.48 in the last reaction means that there's approximately 50% bond formation taking place. So chemists can make reagents with different structures, measure these Brønsted beta values, and find the degree of formation of bonds in a transition state. And these beta values are the only experimental means of determining the structures of most transition states. They're the bread and butter of classical physical organic chemistry with covalent interactions. But in biology, the noncovalent bond dominates so many processes. It dominates some elements of enzyme catalysis. Noncovalent interactions dominate mainly the folding of proteins, the pairing of DNA strands, enzyme substrate recognition, antibody-antigen recognition. All of those are dominated by the noncovalent bond, hydrophobic interactions, hydrogen bonding, electrostatic effects. It's a different game, but it's interesting. After my work on the hydrolysis of aspirin, I was recruited at the MRC Laboratory of Molecular Biology as a mechanistic chemist to work on the mechanisms of enzymes that they were solving, the crystal structures in the lab. And my next mentor was Max Perutz, and here I have a photograph I took of him holding a model of hemoglobin, which was his favorite photograph, I'm pleased to say. And back in 1974, I wrote a paper on catalysis, binding, and enzyme substrate complementarity to analyze how binding energies could affect enzyme catalysis, and it was contributed by Max to proceedings of the Royal Society. It was very easy to theorize, work out equations, and predict, but it was very difficult to do experiments. But this became possible with the advent of site-directed mutagenesis, introduced by Clyde Hutchinson and Mike Smith, whom I'd both met in the MRC Laboratory of Molecular Biology when they were both on sabbatical there in the 70s. Their landmark paper, for which Mike Smith received a Nobel Prize, was published in 1978. Surprisingly enough, despite the obvious power of being able to mutate individual residues in proteins using oligodeoxynucleotides to change the code at various positions, as Greg had mentioned, it really hadn't been taken up because I don't think people had really realized the power of such a procedure. And it was really to our surprise that Greg and I published the first paper of making targeted mutations in a protein of known structure, the tyrosine tyrosine synthetase, which Greg had sequenced. And together, we worked on protein engineering of the tyrosine tyrosine synthetase, and some of the early results were that we were able to measure the empirical strengths of the hydrogen bond in binding specificity, which is one of our most cited papers. We discovered that the mechanism of this enzyme involved just the use of binding energy and no other catalytic groups on the protein, which was really quite startling. But importantly, for what I'm going to talk about now, I was able to show that non-covalent binding energy changes in catalysis could follow the same structure-activity relationships as in the Brønsted analysis. And this was done by, as Greg mentioned, we knew the structure of the intermediate tyrosine adenylate bound to the protein. It made lots of hydrogen bonds between side chains and hydroxyl and other groups on the intermediate. These were trimmed back by Greg with his skills, and we mutated these residues, so we removed specific interactions, and from these, we could start analyzing the change in binding energy along the whole reaction profile. And for example, for the key step, we found that there was a Brønsted beta of 0.76, showing that 76% of the binding energy change was being realized in the transition state of the reaction and not the ground state. This set up the procedure for analyzing protein folding pathways. showing that we could mutate individual side chains in an enzyme, measure the kinetics, et cetera, and have structure-activity relationships like Brønsted, meant that we could do this as a protein folded up by mutating residues systematically over the protein in the same way as a physical organic chemist would make small changes in the structure of covalent molecules to do Brønsted type analyses. And we did this. It was first called beta for binding, like really in honor of beta for Brønsted, but beta goes nicely with binding as well. We make sensible mutations around the protein. We measure changes in activation energy, divide those by the changes in the free energy of folding of the protein, and from that we get this beta value, which for folding we now call phi for folding. So we can make this parameter phi by mutations all over the protein to work out what's going on during the folding mechanism. And I can illustrate this in a sort of simpler way. Supposing we have a small protein which has an alpha helix docked onto a strand of beta sheet in the middle at the bottom, and we look at its folding going from its denatured state on the bottom right, make a mutation in that protein of, say, an alanine, which is in the helix, mutate it to a glycine. We can measure the change in kinetics of the protein as it folds up. If the transition state, as on the far right, has that helix totally denatured, as in the denatured state, then that mutation makes no change in the kinetics of folding, because it's going from denatured ground state to denatured transition state. If that alpha helix is fully formed in the transition state, and we destabilize the protein by, say, 10 kilojoules per mole, by making that mutation, it will destabilize the transition state by 10 kilocalories per mole and slow down the reaction. And that will give us a phi value of 1. So we go around the protein, we make these mutational changes, and we have a phi value of 0, meaning that there's no structure at a particular position during folding. Or phi equals 1, there's complete native structure in the transition state. And then we can get intermediate values, which are most frequent. It's a bit like determining an NMR structure, if those of you have done it, using NOEs. We have weak phis of around 0 to 0.2, little structure, phi between 0.3, 0.6, these are arbitrary boundaries. There's medium, significant structure, and then phi values of, say, greater than 0.7, this is an arbitrary limit. We get strong, highly structured regions in that position. So this was pioneered on a protein called barnase, which had a couple of different domains in it, but it really came into its most powerful force on a small protein called chymotrypsin inhibitor 2, the work done by Sophie Jackson in my lab. And what she found was that this single domain of a protein, only having 64 residues, folded and unfolded by very simple kinetics without an intermediate. It was the first example of a protein folding without an intermediate. And this meant that nucleation mechanisms could perhaps be involved after all. Since then, many, many domains have been found to fold without intermediates. And in fact, this protein folds by a nucleation mechanism, which is a novel one, which we called nucleation condensation. And the reason why we called it that was there's a nucleus of an alpha helix formed, and the rest of the structure appears to condense around that helix. But it's not a tight helix, the nucleus is diffuse and extended. And it has 5 values which are high, but apart from one position, never reach 1. And the 5 values decrease the further and further away you get from that nucleus. And it's like the protein is folding around a nucleus which is being formed at the same time as the rest of the protein is collapsing around it. And there's a very good reason for this physically, and that is for most proteins, their individual elements of secondary structure aren't stable in the absence of the rest of the structure. Protein folding is very cooperative, and in order for a helix to form, it has to be stabilized by making interactions elsewhere in the protein, which we call long-range interactions. And this mechanism has been found subsequently in many places. And this comes to the next point about combining this with molecular dynamics simulations. Molecular dynamics simulations apply Newton's laws of motion to the folding of a protein. The potential functions that are used are good enough in some cases and to do many types of calculations, but they're not known well enough to be able to use them reliable for protein folding and unfolding. But what we could do, once we've determined our 5 values, is for theoreticians to use those to benchmark their simulations. And I had a very long collaboration with Valerie Daggett in Seattle, who do simulations on our proteins without knowing our experimental data. This was very important. There were theoreticians who would only do simulations only if they knew the results in advance, but Valerie would never do this. So we were able to collaborate on a basis of we do the experiment, she do the simulations, and then we put things together. And from this, she was able to fill in the rest of the protein folding pathway using simulation. Our transition states were the benchmark, and we knew the final folded structures, but we could then know the final full pathway. This work proceeded, and we were able to find other mechanisms of folding and then generalize some mechanisms of protein folding for small domains. A protein we looked at soon afterwards was another small protein. It's a helix turn, helix motif DNA binding domain, the engrailed homeodomain, which is a member of the homeodomain superfamily. Just 3 simple helices. And most surprisingly, we found it folded by a pure framework mechanism. The denatured state of the protein has helices in it which are well formed. We could chemically synthesize those helices and find they were 30 to 50% folded in the absence of the rest of the protein. They would then dock to some extent to form a rather helical folding intermediate, whose structure we could determine directly by NMR. We could determine the structure of the transition state very well with our 5-value analysis. We knew the structure of the final folded protein, and we have this whole protein pathway determined, and this protein actually folds on these tens of microseconds time scale. So why do we have this framework mechanism? It's because these helices are overstabilized. They're unnaturally stable, and as part of their biological function, because this protein has to fold and unfold, I think, in its searching on DNA. And this superfamily of homeodonames has led to a unification of mechanisms. We found framework for this highly stabilized alpha helical protein as opposed to the nucleation condensation mechanism for CI2. We worked on other members of the family which have really totally different sequences but the same overall fold. One of them is CMIB, another was HTRF1. And we found that there was a gradation of mechanism. CMIB folds by two-state kinetics, and the 5 values for its transition state ensemble resemble those of CI2. And we were able to get this and other protein in between. And the difference is in the stability of those individual helices. In this graph in the middle, you can see the circles when you plot the helical propensity against the residue number. And the circles are for engrailed homeodomain. So the helical propensity of the first helix is getting up to close to 70%, the second helix 30 or so percent. And then for other members of the family, the propensity gets lower and lower. And this change in helical propensity as we go down the series mirrors a change in mechanism as we go from framework to nucleation condensation. There's a slide of mechanism. There's a folding intermediate for engrailed homeodomain, and it slowly disappears with the others. And so in terms of free energy profiles, if we go along a reaction coordinate of going from the unfolded or denatured state on the extreme right of me to the intermediate state, which could also be a denatured state, to the transition states, and to the native state, engrailed homeodomain at the top has very stable helices, and they can form a stable intermediate with a dip at the bottom of the energy profile. As we decrease the stability of the helices, we still have a pure framework mechanism, but the intermediate gets of higher and higher energy, and the kinetics will tend to go from an intermediate that you see to an intermediate that's higher in an energy pathway, which you can't see. And then eventually, as we get down to a more normal type protein, where elements of secondary structure are rather unstable, we go over to a pure nucleation condensation mechanism. And the 3 classical mechanisms can be viewed as extremes of nucleation condensation. Nucleation condensation, which is, I think, the most common mechanism, is the simultaneous formation of a nucleus and interactions that stabilize it. If we go to an extreme of overstabilized secondary structure, like an engrailed homeodomain, lambda repressor, and other proteins, we get a framework mechanism of where these secondary structural elements will form by themselves naturally and then form the final folded structure. And if we overstabilize the tertiary structure, then we can get that type of mechanism formed. So, is the protein folding in vitro relative to that in vivo? Well, we know that chaperones, molecular chaperones, are involved in the folding of many proteins to stop them aggregating in the cell, and in multi-domain proteins, that's very important. And also, the protein synthesis on the ribosome is vectorial. So, what do we know from the in vitro experiments? Well, the fundamental processes are likely to be the same, and that individual domains will use the same type of mechanisms. The larger proteins, though, will be affected more, ones which fold in various domains and then they have to get together. But with small proteins, they fold so fast that they escape the chaperone system and almost certainly fold the same way in vitro as in vivo. And these small proteins fold immediately on leaving the ribosome, because if they're small enough, too much of them is buried inside the channel of the ribosome for it to fold before leaving. But most importantly, protein unfolding is not affected in the same way by these vectorial processes of synthesis and chaperones. And protein misfolding happens usually via proteins unfolding. In work I'm not going to talk about because I have no time. I've done a lot of work on the tumor suppressor P53, where many of the mutations that cause this key tumor suppressor to be inactive work because they cause the protein to unfold. So, to end up, 5 values per se have shown that the transition states for folding of small domains, these distorted native-like structures, they're extended and stabilized by many of the native interactions, that the mechanisms folding are often concerted, secondary and tertiary, of the nucleation condensation mechanisms, sometimes framework mechanisms, and there's a variation in degree of structure formation from the very diffuse chymotrypsin inhibitor 2 transition state to those strong interactions in engrailed homeodomain, which make a polarized transition state where much of the structure is formed with high values of phi. So phi analysis has really been used quite widely in protein folding studies, and I just list here, don't take any notes of the individual examples, just phi analysis with other proteins. Many have been done, which are used to sort of generalize processes, and phi values are used in other systems, the folding of transmembrane proteins, in receptors and gating, and interestingly, for the folding of intrinsically disordered proteins, which fold when they bind to folded partners. I'm very pleased to say that much of that work has been done by former workers from my lab, and proteins where mechanical force mimics their function, and it's been extended to oligonucleotides for RNA folding and DNA aptors. Thank you so much for listening to me.